Welcome back. So in some of the last videos, we looked at protective organelles, basically from the cell's perspective, and those were the peroxisomes, which protect the cell against hydrogen peroxide. And then we looked at lysosomes, which are oftentimes contained in cells like white blood cells, and they can protect against foreign proteins and bacteria. But we're going to switch gears a little bit, and we're going to talk about a different organelle that is of astronomical importance in most cells of eukaryotes. There are a few exceptions here and there, like red blood cells, but in general, the vast majority of energy that is produced by the cell is done by this organelle right here, referred to as the mitochondria. Okay, now before I go any further, I want to be sure that we understand a few things. Number one, if you're watching this in the context of biochemistry, this particular video probably won't help you a whole lot. We're really just going over mitochondria anatomy and some basic functions, but from an A&P, anatomy and physiology perspective, this is going to be perfect. Also, the other thing that I want to go over is basically what ATP is, okay? ATP, number one, is a nucleotide, and what ATP stands for is adenosine. It's adenosine triphosphate, okay? And here's the idea behind ATP. You have various processes in the cells of eukaryotes, and prokaryotes for that matter, that require energy input. Okay, so a lot of biosynthetic reactions. In fact, we already looked at one that needed it. When we looked in previous videos at the function of aminoacyl tRNA synthetase, we essentially found that that particular enzyme required ATP. And actually the reason it required ATP is because you have to input some energy into the reaction to get it to go to completion. So the enzyme can't do it without the use of ATP. So what ATP essentially is its energy currency. That's a common term or phrase that people use to describe ATP. So essentially what it is it's like energetic money from the cell's perspective. So you have various processes in cells that produce ATP. That's like making money from the cell's perspective. Then you have some processes that's like spending money, where you have to burn, or in other words, you burn an ATP or you use the ATP. Sometimes I use the term burn ATP. That's pretty typical when you're talking in biochemistry. But the idea is that ATP provides energy for reactions to go to completion. And the vast majority of ATP is created in this organelle known as the mitochondria. And I want to be very specific about this. Remember that prokaryotes don't have membrane-bound organelles, and this is certainly a membrane-bound organelle. So this is exclusively a eukaryotic organelle, because as we said, prokaryotes don't have those. Okay. So let's go into some basic structure of the mitochondria. So this right here, this organelle that's shown right here, if I can find my mouse, here it is. This organelle right here is known as the mitochondria. And you should notice that it has two membranes. So number one, if you look at it, here's the outer membrane. So let me kind of highlight that. This is the outer membrane as shown right there. And then I'll use in blue to show you the inner membrane. So the inner membrane is this one in here. If you look at the space that's between, oops, that didn't go through, let me re-highlight that. If you look at the space between the inner membrane and the outer membrane, this is what's referred to as the inner membrane space. So all this business in here, this space in here, this is referred to as the inner membrane space. Now, there's actually space within the inner membrane itself, okay? So all this space that's in here, this is actually confined within the walls of the inner membrane. And so there's also various processes that take place inside there, as we'll find things like beta oxidation and the Tca cycle occurred there as well. Okay? And one thing that's also really important to understand is, I'll do this in green, is when you look at the membrane, the inner membrane, what you'll have here are various enzymes that exist in the membrane. Okay, so these are membranous enzymes. They exist in the inner membrane of the mitochondria. See, why did it do that? It deleted that. Anyways, but you have various enzymes that exist in the inner membrane of the mitochondria, and they're vital for energy production. Okay. Now, oftentimes what you'll see is this term known as cristae. Okay. The singular word is crista. Okay. What a crista essentially is it's a component of the inner membrane that has these branches. So the actual branches and the bending in this inner membrane are described by the crista. So crista basically is this situation in which you have the bends right here. See how the inner membrane is kind of bending and snaking around, right? That whole business where it's kind of snaking around and bending and giving these invaginations, that's what a crista is. And the entirety of it is plural cristae. Okay? So literally the cristae is just a term to describe the inner membrane. Okay? So the invaginations and the bends and the snaking around of the inner membrane, that's the cristae. Okay? Now, when you look inside the inner membrane, that space in here that I designated, what did I designate it as? I designated it as black right here. This space in here, okay, this is known as the matrix. Okay, so we have a lot of metabolic processes like the Tca cycle that occur in the matrix of the mitochondria. Okay, and one thing that's also important to realize is the nucleus of the cell is not the only place where you'll find DNA. In fact, the mitochondria also has its own DNA, and it also has its own ribosomes. Okay, that's important to realize. What also is really important is to have a basic understanding of some of the really important processes that go on inside the mitochondria. Of really important note is this very general function, generation of massive amounts of ATP. So all of these cellular processes that go on, you have massive productions of ATP relative to that is produced in the cytosol. So cytosol would be like where glycolysis occurs. Glycolysis only produces about a net 2 ATP. Okay, that's not a lot. Whereas if you look at one complete catabolic pathway for glucose in the mitochondria, that produces more like 30 to 32. So there's much more ATP that's produced inside the mitochondria than there is in the cytosol. Okay? There's also a pathway known as beta oxidation. This is the catabolic pathway for fatty acids. Very important from the liver's perspective in times of fasting. And if you get excess fat from the diet, it'll be beta oxidized. Another energy-producing pathway is called the Tca cycle, or sometimes you'll hear it referred to as the Krebs cycle. This also produces energy, and it's located in the matrix of the mitochondria. And then the whopping amount of ATP that's produced, do this in another color, this is by the electron transport chain. Sometimes this pathway is referred to as the respiratory chain, or the respiratory chain. Okay. I sometimes call it the respiratory chain. Understand that I mean the electron transport chain. Usually people abbreviate it ETC. That's pretty common. There's also another really important pathway that goes on here, and that's Hien Biosynthesis. So in humans, we synthesize a molecule called heme, and this term porphyrin here, that just porphyrin is a class of molecules, and heme is going to belong to that class. But the idea is that humans, in the mitochondria, part of the synthesis of heme occurs there, and we'll talk about the implications of that. Okay, we're not going to go into any heavy-duty pathways or anything. Okay, so like I mentioned, mitochondria have their own ribosomes and DNA. That's important. Okay, so this is where we get to the first pathway, and by no means do you have to memorize all this. There are some things I want to point out that are important that you do need to know. Okay. There's this molecule right here, and it's vastly abbreviated because it's actually a very large molecule. This molecule is called acetyl-CoA. Okay, and let me do this. Let me denote. So these are the two membranes in the mitochondria. This is the matrix. This is the got an email. Sorry. This is the matrix of the mitochondria. Okay. Out here, I'm going to assume that this is the cytosol. Okay. And in the cytosol, we have the process known as glycolysis. So what will happen is glucose, through a multi-step pathway, will get converted to a molecule called pyruvate. Okay? Pyruvate's a critical molecule because it's going to be essentially transported into the mitochondria, and pyruvate's going to react with an irreversible enzyme, you don't need to know this, called pyruvate dehydrogenase, or PDH. And PDH is going to convert pyruvate into acetyl-CoA. So ultimately, the carbon fragments that come from glucose that you get from the diet or from your liver, the carbon fragments of glucose form pyruvate. The pyruvate gets transported into the mitochondrial matrix, and then pyruvate dehydrogenase converts it to acetyl-CoA. And oftentimes, this acetyl-CoA, we refer to this as the central coenzyme in all of biochemistry and molecular biology. Now, this cycle is called the citric acid cycle, and sometimes you'll hear it referred to as the Krebs cycle. Sometimes you'll even hear it referred to as the Tca cycle. Either one of these three names will work on an exam. But what I want you to realize through this cycle is we produce several things. Number one, notice that in this reaction right here, in this reaction right here, and then this one way up here, what do we produce? Well, overall, we produce 3 NADH, and we'll talk about that in a minute. And then, through this reaction right here, succinate dehydrogenase, you don't need to know that, we produce something called FADH2, okay? These two molecules, NADH and FADH2, these are what we call reduced coenzymes. Sometimes you hear it referred to as reduced cofactors. This NADH is going to play a role in energy production by the mitochondria, i.e. the electron transport chain, and so is this FADH2. So what you need to know is simply that this pathway, along with the glycolysis and the cytosol and beta oxidation in the mitochondria, produce, highlighting, NADH and FADH2. NADH and FADH2 are energized coenzymes that will use their energy to power the electron transport chain in the mitochondria. That's their very critical function, and we'll talk about the implications of that in a few minutes. But one thing that you do need to be aware of is you do need to know these two coenzymes, which are NADH and FADH2. Now, the nice thing about the citric acid cycle that's good is the same coenzymes are produced in beta oxidation. So we're going to look at beta oxidation in just a minute. We'll come back to that. But the same coenzymes are produced here. In fact, let's do this slide really quickly. Okay? Beta oxidation is the catabolic pathway for fatty acids, and it leads to energy production. So what I want you to realize is the following. Number one, this first enzyme right here, what does it produce? An FADH2. Okay? This second enzyme right here, let me do it in green, this enzyme produces NADH, okay? And then, of course, at the very end, we produce acetyl-CoA. And that acetyl-CoA, notice, can go into the Tca cycle. So not only can we produce acetyl-CoA through this pyruvate oxidation to acetyl-CoA, we can also produce acetyl-CoA through beta oxidation, and this will essentially go into the Tca cycle. But what I'm concerned about is this FADH2 right here and this NADH, okay? So let's talk about what happens to those guys, okay? So here's the basic idea, okay? NADH and FADH2 generated by glycolysis, the Tca cycle, and beta oxidation fuel, this is important, fuel the pumping of protons by enzymes in the inner membrane of the mitochondria. Okay, so here's what you need to kind of be aware of. Okay, in here, notice this is the mitochondrial matrix. Okay, this right here is the inner membrane space. Okay, what I want you to know is that what NADH right here, what NADH and of course FADH2 do effectively is through a series of processes, okay, NADH does it directly, FADH2 does it indirectly, but essentially what they do is notice the direction of this arrow. They're pumping protons from the matrix into the inner membrane space. Let me be specific. So protons, or H+, in the matrix are pumped into the inner membrane space. Okay? Now, if you think back to your cell physiology, well, If I'm pumping lots of these protons, these H pluses, from the matrix into the inner membrane space, that's going to essentially create a large amount of protons out here. So I'm going to develop a highly concentrated region of protons out here in the inner membrane space, and what that's going to do is create a concentration gradient. And so basically what you want to know is these protons are essentially going to come back here through ATP synthase, and they're going to go back into the mitochondrial matrix, and they're going to power the synthesis, I'll do this in a bold color, of ATP. So what is the enzyme that synthesizes ATP? It's called ATP synthase. So ATP synthase is the enzyme that biosynthesizes ATP, and the way it really works on a mechanical level is the protons that are pumped into the inner membrane space by these enzymes over here, the protons essentially rush back through and cause mechanical energy that's used to synthesize ATP. That's what you need to know. Okay, so hopefully you can put that in your own words if I ask this on the test. Okay, let's test ourselves. Which of the following metabolic pathways does not occur in the mitochondria? Well, we just showed this electron transport chain. Notice the electron transport chain occurs in the inner mitochondrial membrane, right? So electron transport chain is not the answer, so that's out. Glycolysis, well, if we go back to this slide up here, I mentioned glycolysis actually does occur in the cytosol. Okay, so glycolysis in this case is the answer. This is not in the mitochondria. But let's also look at these other pathways. Go back to beta oxidation. This also occurs in this will occur in the mitochondria. Most of these enzymes are in the mitochondrial matrix. And then the other answer is the Tca cycle. That also occurs in the mitochondrial matrix. So, which of the following does not, pay attention to this, does not occur in the mitochondria? It's glycolysis. This occurs in the cytosol. Okay? Next question. What are the products of the Tca cycle that power proton pumping by the electron transport chain? And I could essentially add on to this that powers ATP synthase, right? Make sure you keep that in mind. The proton pumping by these enzymes fuels ATP synthase, and ATP synthase will essentially spin around. It'll rotate around and around and around, and that spinning generates ATP. So the concentration of ATP will essentially sharply rise. Increased ATP by ATP synthase. So what are the two products of the Tca cycle that power proton pumping? Well, let's go back to the picture. So what two products are there? Well, I highlighted those. Those are going to be NADH. We generate 3 of them per cycle, and then 1 FADH2. Okay? So the answer in this case is going to be NADH and FADH2. Okay? And there's just one more thing basically to go over. There are a lot of enzymes and proteins in biological systems that require this molecule right here. This molecule is called Heme B, and the key is that it's a heme. Hemes belong to a class of molecules known as porphyrins. Porphyrin is the singular version of the word. And Heme B is just one of these porphyrins. There's also a Heme A, not shown here. There's also a Heme C, a Heme O. Basically, the idea is there's lots of hemes, and these are used by proteins and enzymes to catalyze reactions and do other things. For example, hemoglobin, thus the name heme, is the transporter of oxygen in your blood. So whenever you take an inhale and you inhale gas, there's oxygen there, and the oxygen will essentially cross from the air in your lungs into your blood and be picked up by this transporter of oxygen known as hemoglobin. So it uses actually this iron in the center of the heme to actually bind the oxygen. Okay? Also, there are enzymes that also use this coenzyme. Okay, so this Heme B, one thing to realize, it is a coenzyme. Okay, it's used by enzymes. And some of the enzymes that use it are catalase, which we have talked about previously, cytochrome P450, which we also talked about in the past. Okay, and the way that this heme essentially works is it catalyzes or allows the catalysis of one electron transfers. Okay. part of the synthesis of this molecule is done in the mitochondria. Okay, that's all that I care that you know. Not all of the synthesis is done in the mitochondria. Some of it's actually done in the cytosol. But approximately half of the synthesis is actually done in the mitochondria, making mitochondrial function absolutely required for porphyrin synthesis. Actually, what's interesting to note is there are very common, I wouldn't say very common, but relative to other diseases, they're kind of intermediate in commonality. There are deficiencies of porphyrin enzymes. So there's actually, I'd have to look back, I think there's 8 enzymes in heme synthesis. And if you have a deficiency of enzyme in Heme B synthesis, if you have a deficiency of one of these approximately 8 enzymes, this is referred to as a porphyria. Okay, this is a porphyria. And porphyrias are basically the origin of the vampire stories, basically. So the story goes that there was a guy he was kind of royalty, basically. And basically, a lot of times, when you have porphyrias, meaning a deficiency of enzyme in heme synthesis, you crave blood. Okay, so what this guy would do is he couldn't be out in the sun, because when you have porphyrias, you have all sorts of skin problems. You have sensitivity to sunlight, and so really the best time for you to be out is when the sun goes down at night. And when you have porphyrias, a lot of times you crave meat and blood and things like that, because humans are actually able to sense that blood and meat actually have lots of heme in it. Okay, so he would crave this stuff, and at night he would basically go outside and go to farms, and he would kill animals and drink their blood. And that really right there is the origin of where vampires come from. And on top of that, what some porphyrias do is they give you things like sharp teeth. They change the color of your eyes and things like that. So he would have been perceived as being the prototypical vampire, basically. Okay, so that's the origin of it. When you have a deficiency of the enzyme that synthesizes this guy, that's called a porphyria. And some of the porphyrias are located in the cytosol because some of the enzymes are in the cytosol. But about half the enzymes are in the mitochondria, and so if you have a deficiency of any of those enzymes, it's called a porphyria. Okay, so hopefully this video gave you a little bit of an intuitive sense about mitochondrial function and structure. Make sure you can, for the practical part of the test, let me star this. For the practical part, anything that I labeled on here, probably with the exception of the DNA and the ribosomes, because you can't really see that well. But if I point to, say, the matrix space, or the cristae, or say the outer membrane, inner membrane space, if I point to any of that, make sure that you can identify what structure I'm talking about. That would appear, for example, on the practical part. And then for the function, just make sure you understand what pathways occur in the mitochondria, okay? And kind of be able to give a general function of them. Notice that the Tca cycle, the Krebs cycle, whatever, and beta oxidation, notice that they produce both FADH2 and NADH, the reduced and energized coenzymes. And then those coenzymes are used to power the pumping of protons. Notice the pumping of protons from the matrix into the inner membrane space, and that powers ATP synthase for the synthesis of ATP. If you can basically say all of that in your own words, or even in those words, to be honest, you'll get full credit on that question on the test. There is an essay in your practice problems where I ask you about the mitochondria. It's possible that will end up on the test because, quite honestly, the mitochondria is a really important organelle in eukaryotes. Okay, so hopefully this gave you a little bit of intuition on the mitochondria. See you in the next video.